Hey guys, so this is the video for chapter 11. In chapter 11, we discussed meiosis, and I already have a video made on meiosis about the steps of meiosis, why each one is important, and we will talk about later in this chapter about sexual versus asexual reproduction, but definitely understand the process of meiosis, how it's similar to mitosis and how it's also different from mitosis. Know that meiosis is only used on reproductive cells. Meiosis isn't happening throughout the body. This is only happening to make gametes. That's the whole purpose of meiosis, is to make gametes, is to make sperm and eggs, as far as us, as far as humans. So mitosis happens on all the cells in the body, the regular somatic cells in the body. But if we're talking about making sperm, if we need to make eggs, that's meiosis. If you got to make gametes, you undergo this chapter, you undergo meiosis. So you would go from our diploid state right now to make a haploid gamete that's got half your chromosomes, and it's going to meet up with another gamete that's got half the chromosomes, and they will come together and form a diploid organism for us. So know your phases of meiosis. We've got the first picture here talking about interphase, that most of the cells we just discussed are in interphase for 90% of its life. Important parts of meiosis, we talked about it in the previous video, but know what happens in prophase 1, that it's crossover. It's the most important part of meiosis, because without crossover, you're not getting that genetic diversity that you need to make sure that this sperm isn't the exact same as that sperm, and that egg doesn't look exactly the same as a previous egg, that all of these things, these gametes that are produced, are genetically unique, and that wouldn't happen unless you had crossover happening at prophase 1. So one of the most important steps. So here's crossover picture happening, prophase 1, prophase 1. The law of independent assortment is when we are, remember how we talk about in metaphase, whether it's metaphase 1 or metaphase 2, but no matter what happens in metaphase, it starts with an M, and that means the chromosomes line up in the middle. And the law of independent assortment says that it doesn't matter which side, right side or left side, what side of the cell it's going to line up at, it's got a 50-50 shot of whatever you have going towards one side or the other. That's the law of independent assortment. The way it lines up at the middle, it could line up on either side of the middle depending on what's going to go to the right side or the left side, so to speak, of the cell. That's the law of independent assortment. It's going to be random 50-50 shot. I'm going to skip through. This is the table that summarizes all the steps. Again, we talked about it in the previous video, so please watch the video talking about meiosis and comparing meiosis versus mitosis. Meiosis, genetically unique. Mitosis, genetically identical. Meiosis is going to go from a diploid starter stem cell, and it's going to end up with a haploid gamete at the end. And you may have one if we're talking about eggs, or you may have 4 if we're talking about sperm. But you're going to end up with one haploid gamete egg at the end when you start off with a diploid egg or sperm stem cell. So you go from diploid to haploid. When you're talking about mitosis, it's diploid to diploid. You just make a carbon copy of your cell. Everything's the same. Everything's genetically identical in mitosis, but meiosis is going to be unique. That's what's so cool about meiosis. Everything's unique. Every sperm is different out there. Every egg is different out there. So definitely know the steps, know what's happening in the steps, be able to compare and contrast mitosis and meiosis in chapter 11. I'm going to move on to the rest of the chapter that did not get covered in the previous video, and that's going to be talking about cellular reproduction and why you would even want to undergo this process. Will I get it to work for me? Yes, it is working. So why would you want to do this? What are the pros and cons of sexual reproduction versus asexual reproduction? Asexual reproduction, easy, so easy. We talk about budding, or a little fragmentation, a piece of you breaks off, and then that thing grows up into a whole nother you. It's super easy. It's not a whole lot involved to break off a piece of yourself and that thing grows up just like you. Asexual reproduction is easy. That's one of the greatest benefits of asexual reproduction. One of the cons, though, is everything's just like you. Everything has your genes, and if those genes predispose you for one thing, and you're very specialized, and the environment changes, something happens, you're not ready for it. If everybody is exactly the same and something comes in that's a danger or a threat to you, if you don't have the genes that are adaptive for it, the entire population gets wiped out. So genetic diversity is a benefit of sexual reproduction. That's the biggest benefit of sexual reproduction, biologically speaking, is genetic diversity. Cons of sexual reproduction: it is labor intensive. It takes energy to reproduce gametes. It takes energy to make gametes. It takes energy to make yourself attractive to find a mate. Whether you are a person, whether you are an animal that has to go out there and find the mate. If you were trying to, say you were a grizzly bear and you had to go walk miles and miles and miles to find a mate, and then maybe you had to fend off that mate from other competitors that try to steal your mate. All of this is energy spent on sexual reproduction. If you are a flower, you have to grow those pretty petals and attract the bees so that they will pick up the pollen and go pollinate the other plant. It's labor intensive, it's energy intensive. Those are the cons of sexual reproduction. But the benefit is that your genetic diversity for your offspring will be hopefully better, with more distinguishable traits that will set you off and differentiate your offspring from others. And that variety of traits we talked about in natural selection and evolution, we're going to get to that. Having a variety of traits means that you're hedging your bets, that some of those offspring will be better off than others, and some of those offspring are going to make it. Genetic diversity is key. So that is the benefit of sexual versus asexual reproduction. Life cycles. There are different types of life cycles. Oh, I forgot about the quote from the Looking Glass from the textbook. Basically saying that if you're not adapting, if you're staying in one place, you aren't going to make it. Sorry, my dog is doing crazy zoomies in the background. You can hear that. She's growling at invisible devils right now. I'm sorry. Okay, back to, if you're not going to change, if you're not able to adapt, you will not make it. Life cycles, different life cycles. There are three that you're going to need to know, and you're going to need to know the different types and examples of each one. So diploid dominant, that's us. We are the example for this type of lifestyle, diploid dominant. So we said our meiosis is only used to make gametes. We're only using meiosis to make sperm, to make eggs. Those gametes have half the genetic content. So they are haploid. Haploid is 1N. So you're going to have 1N here, and you're going to have, it's going to meet up with the 1N from the sperm, and they will fuse, and now we have a zygote that is 2N, or diploid. So our diploid cell is now going to undergo mitosis, 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 and that one cell, that one diploid zygote, is the stem cell for all the other cells in your entire body. Come from that one cell that happened when two haploid cells fused together, and they fused their genetic information and became one diploid cell. And so we undergo mitosis, 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 and our life cycle is dominated by being diploid. Our cells are diploid when we are multicellular, versus fungi. Fungi is an example for haploid dominant life cycles. So when they are multicellular, they're multicellular at their haploid stage. They are going to undergo meiosis to produce spores. They will grow, and I like to say they hatch, because it's kind of like an egg. Stop it. Stop. The spores are going to, I'm so sorry. The spores are going to grow, and they are going to grow and develop and undergo mitosis, and they are going to develop into fungi. Fungi have these little roots called hyphae, and they're not, you can't call them roots because they're fungi and they're not plants, but the hyphae are kind of like roots for fungi. And so all these cells, when they're multicellular like this, they're all haploid. They have half the genetic content, and that's okay with them. They're multicellular at this stage. And so then this haploid hyphae is going to meet up with this haploid hyphae, and they are going to have cells that come together and they will fuse. We'll talk about karyogamy when the plasmogamy when the cytoplasm fuses, and then karyogamy when the nuclear fusion happens. So nuclear fusion is going to happen. It's going to make a zygospore. The zygospore is diploid. That diploid zygospore is going to undergo meiosis. And what we know from meiosis is that you end up with a haploid offspring. So these guys, this is their huge multicellular lifestyle. And when they are multicellular, they're doing it at the haploid stage. So an example for haploid dominant life cycles are fungi. Diploid dominant life cycles, us. We are multicellular at diploid stage. Fungi are multicellular at the haploid stage, and plants are multicellular at both. Plants are going to be multicellular at a sporophyte stage, and plants are going to be multicellular at a stage called a gametophyte. Sometimes they will be more dominant at one or the other. Like you'll notice a fern is the example for the sporophyte here, or pine trees. Maybe you might see the sporophyte as the typical dominant part of that plant, the pine tree, but there's also a gametophyte part of that plant that is multicellular, like the pine cone, or like this guy right here with the liverwort. Um, it's also multicellular. So it's not just multicellular at one stage or the other. It's both. It's multicellular when it's diploid, and it is multicellular when it's haploid. So plants are weird, they're unique like that, and they have alternation of generations. They are multicellular at both diploid, and they are multicellular at the haploid. The diploid stage is called the sporophyte. The haploid stage is called the gametophyte. In 1108, you'll go through this way more in depth, an alternative of generations, when you go into the plant section and all those chapters later on in the textbook. So you'll go through this more in 1108, but if you're curious about alternation of generations, there is a video that Miss Rogers did, and I attached it into the D2L course page, so you can watch that if you're curious. I need you to know for this class right now what diploid dominant means, that you're multicellular at a diploid stage, and what's an example of it? Us, me, you, diploid dominant. Haploid dominant, you are multicellular at the haploid stage. An example of haploid are fungi, bread molds, mushrooms, those guys, haploid dominant, multicellular. Plants are going to be an example of something that is multicellular at both. Since they are multicellular at diploid and they are multicellular at haploid, they alternate between the two, so it's alternation of generations. So know the three styles, know what they mean, and know the examples for each of them at this chapter. So that concludes what I need you to know for chapter 11. Stay safe, wash your hands, have a good one.